The South Atlantic Red Snapper Research Program brings together scientists from universities and state and federal agencies to study red snapper within the South Atlantic region of the U.S. The project's goal was to estimate how many red snapper, aged 2 years or older, live from North Carolina to the Florida Keys using two methods: close kin mark recapture and Bayesian hierarchical integrated modeling. The first method, close kin mark recapture, is based on the idea of tagging and recapturing fish, but instead of using physical tags, it uses each snapper's unique DNA as a natural tag. DNA from each individual was collected via a small, non-lethal fin clip sample. Scientists used research surveys and worked with commercial and recreational anglers to collect more than 14,000 fin clips for this study. Samples were collected from 2021 to 2023 to estimate the population size of South Atlantic red snapper. By looking at similarities in each fish's DNA, scientists can determine which ones are related, like parents, siblings, or children. If they find only a few related fish, it likely means a larger overall population size. If they find a lot of related fish, the population is likely smaller. Scientists determine the age of each fish by counting growth rings formed in ear bone structures called otoliths, and by analyzing age-related changes in the fish's DNA. Put all together, the close kin mark recapture method can provide accurate population estimates when information about the fish's age, sex, and reproduction is available. The second method, the Bayesian hierarchical integrated modeling, combines prior knowledge with new data to strengthen estimates. For this study, scientists used data from the Southeast Reef Fish Survey, which has sampled red snapper populations since 2011, and combined this with the use of underwater cameras and remotely operated vehicles to count red snapper at random sites from North Carolina to Florida. The remotely operated underwater vehicle camera footage helped identify red snapper in new areas that the Southeast Reef Fish Survey may not typically sample. By estimating fish population density and combining it with seafloor depth and habitat data, scientists modeled the total number of red snapper at local, state, and regional scales. Throughout the project, the team worked carefully to account for uncertainty, or things that could affect the accuracy, to make sure their population estimates were as reliable as possible. To read the full report and learn more about the South Atlantic Red Snapper Research Program, visit sarsrp.scseagrant.org.